Hey everyone, and welcome back to the deep dive.
Hello.
Today, we're sort of unlocking a secret world. It's hidden right there in plain sight. How plants actually experience the light around them.
hmm
And forget just needing light for energy, like plugging into a socket. Plants are these incredible light detectives. They use light not just to survive, but to, well, know where they are, what they should be doing, and when to do it.
That's absolutely right. Light is so much more than fuel for a plant. It's really, it's a communication system. It carries this vital information about their environment. And that information triggers huge changes in how they grow and behave.
And we've got this fantastic guide for our deep dive today. It's chapter 16 from the 6th edition of Plant Physiology and Development.
A classic text.
Yeah. Now, I know it's a textbook chapter, but honestly, it's just packed with these mind-bending discoveries about the plant's hidden sort of visual system.
You could think of it as the user manual for how a plant makes sense of light. It reveals all the molecular players involved. It's quite detailed.
So, our mission for you today is, well, to unpack this chapter. We're going to zero in on the most important ideas about how plants sense light, what these light sensors, they're called photoreceptors, what they are, how they work, and, why it's all just so crucial for a plant's life. We'll pull out the surprising nuggets that really make you see plants in a whole new light.
It really is about understanding the sophistication behind what seem like simple plant actions.
Okay, let's unpack this. So the first big idea, right, is that sunlight has this dual role for plants. One, it's energy for photosynthesis, turning light into sugar. We've talked about that before. But two, and this is really key, it's also a signal.
Yes, it's information, pure information. The plant uses the quality of light, the colors, the quantity, so how much light there is, and even the direction the light's coming from to make decisions.
And these decisions are absolutely fundamental to their survival. The chapter gives some fantastic examples, like some seeds won't even sprout unless they detect light. That's called light-dependent germination, or photoblasty.
Right. Or think about a seedling pushing up through the soil. In the dark, it's all pale and spindly, stretching like crazy to find light that's ethylation.
Yeah, I've seen that.
But the moment it senses light, boom, everything changes. Stem growth slows right down, the leaves start to expand, it begins making chlorophyll. That whole transformation is called photomorphogenesis.
I've seen that happen on my windowsill. The difference is stark.
Yeah.
And watching a plant bend towards the sun, that's phototropism, right? Sensing the direction of light.
Exactly. And blue light is the primary driver for that. Plants also sense non-directional light for things like timing their leaf movements. Opening up during the day, that's photonasty, and folding up at night, nyctinasty.
Okay.
And critically, they use light to figure out when to flower based on how long the day is. That's photoperiodism.
Sunlight also has UV radiation, which can be damaging, but plants sense that too.
Yes, they sense UV and trickle in protective responses, like making compounds that basically act like sunscreen for the plant. So sensing light is also about, damage control.
Wow. So all these different responses, triggered by different aspects of light, it really tells us plants aren't just passive. They're actively perceiving their environment using these specialized tools. And these tools are the photoreceptors.
Right. You can think of them as the plant's eyes, obviously not like our eyes with lenses and retinas, but specific molecules inside the plant cells that can catch light. They absorb light energy, and that absorption causes a change in the molecule itself, which then, triggers a signal inside the cell.
Sort of like how a hormone receptor binds a hormone and kicks off a response.
Precisely. A photoreceptor absorbs light and kicks off a light response. It's a very similar principle.
Okay.
And most of these photoreceptors are proteins, usually with a special helper molecule attached called a chromophore. That chromophore is the part that actually grabs the light photon.
Ah.
There is one exception, UVR8, which we'll get to later. It uses part of the protein itself instead of a separate chromophore.
Interesting. And plants aren't sensitive to all colors equally, are they?
No, not at all. They have different receptors tuned to specific wavelengths. Red, far red, blue, and UVB light. Those are the really important signals they're reading.
So what are the main families of these receptors?
Well, the chapter introduces the big ones. First, you have the phytochromes. They primarily sense red and far red light, but they also respond a bit to blue and UVA.
Okay, phytochromes.
Then you have cryptochromes and phototropins. These are the main sensors for blue light and UVA. Cryptochromes are generally more involved in overall growth patterns and timing things like the internal clock,
Right.
whereas phototropins are really key for figuring out which way the light is coming from that phototropism and for optimizing photosynthesis. And there's also the ZTL family mentioned briefly, linked to the circadian clock.
Now, one challenge for scientists must be that plants are usually getting hit with the whole spectrum of sunlight, right?
Right.
And multiple receptors could be active at once. How do they figure out which color is doing what?
That's where a clever technique called action spectroscopy comes in.
I love this. Explain it.
Okay, so you expose the plants to very narrow bands of light, basically, one specific color at a time, and you measure how strongly the plant responds. Maybe its germination rate or how much it bends.
Right.
Then you plot the strength of that response against the wavelength of light you used. You get a graph, the action spectrum, and it shows you exactly which colors are most effective at triggering that specific response.
The classic example is lettuce seeds, isn't it? Red light makes them sprout.
And far red light stops them.
Yeah.
Exactly. The action spectrum for sprouting peaks right in the red part of the spectrum,
Yeah.
and the spectrum for inhibition peaks in the far red.
And those peaks match the absorption of phytochrome perfectly.
Precisely. Is incredibly strong evidence that phytochrome was the photoreceptor responsible. And similarly, for blue light responses like phototropism, the action spectra have these characteristic patterns, sort of like three fingers.
Yeah, I've seen those graphs.
And that pattern matches the absorption spectrum of a part of the phototropin receptor, the LOV2 domain.
So action spectra link specific light colors to specific receptors. What about the amount of light?
Right, quantity matters too. The chapter introduces two key terms here, fluence and irradiance.
Okay.
Fluence is the total number of photons hitting an area. Think of it like the total dose of light. Irradiance is the rate how many photons are hitting that area per second, so how bright the light is.
Got it. Total dose versus brightness.
And understanding this is important because different plant responses need different amounts of light, different fluences or irradiances. And you also have to remember that not all the light hitting the leaves necessarily reaches the photoreceptor. Some gets absorbed by chlorophyll first, for instance.
Makes sense. Okay, let's dig into that first big family, phytochromes. The chapter mentions they're found all over, suggesting they're really ancient light sensors.
They are indeed ancient. Their core function, particularly in plants, is to act like a kind of molecular switch that gets flipped by red and far red light. They exist in two main forms, PR, which absorbs red light, and PFR, which absorbs far red light.
And the cool part is they can flip back and forth.
Exactly. That's the key. Red light hits PR, converts it into PFR. Then far red light hits PFR, and it converts back into PFR. This is called photoreversibility, or sometimes photochromism.
PRPFR.
That's the heart of it. And generally speaking, PIR is the biologically active form. It's the one that sends the signal to the plant to do something.
So more red light means more PFR, which means more signal. More far red light means more PR, less signal.
That's the basic idea. And even under normal sunlight, which is both red and far red, you always have a mixture of PR and PFR because their absorption spectra overlap a bit.
Yeah.
They reach a kind of equilibrium called the photostationary state.
But what's really crucial then is the ratio of red to far red light.
Precisely. Think about a plant under a canopy of leaves. Those leaves above are absorbing a lot of the red light for their own photosynthesis.
But they let the far red light pass through more easily.
Exactly. So the plant underneath experiences light with a much lower ratio of red to far red light.
So the phytochrome system senses that low RFR ratio and basically tells the plant, hey, you're in the shade.
You got it. And that signal triggers things like stretching taller, trying to reach unfiltered sunlight. That's the shade avoidance response.
How does the phytochrome actually change?
It's a protein dimer, so two copies join together. Inside, it holds that chromophore, phytochromobilin. When the chromophore absorbs light, it physically changes shape, a little twist in its chemical structure. This shape change in the chromophore then causes the whole protein structure around it to rearrange.
And that protein shape change is the signal. Where does it go?
Well, phytochrome is made in the main part of the cell, the cytosol. But when red light converts it to the active PFR form, it actually moves into the nucleus, which is, the cell's control center.
Okay, so light makes the active form, PFR, go into the nucleus. What's it doing in there?
Its main job in the nucleus is to interact with other proteins that control which genes get turned on or off. It regulates gene expression. This is how phytochrome triggers those slower, more long-term developmental changes, like seedling development or influencing flowering time.
But the chapter also mentioned faster responses, things happening in seconds or minutes.
Yes, there's evidence for that too. Some rapid responses, like changes in ion flow across cell membranes, might be initiated by phytochrome that's still in the cytosol, perhaps interacting with components at the cell membrane itself.
So nuclear action for long-term changes, maybe some faster stuff outside the nucleus too. Does the amount of light matter for these responses?
Absolutely. The chapter groups phytochrome responses based on how much light they need. There are very low fluence responses, VLFRs, low fluence responses, LFRs, and high irradiance responses, HIRs.
Very low, low, and high. Okay.
VLFRs need incredibly tiny amounts of light. We're talking starlight levels almost. They get saturated, meaning the maximum response happens with very little light. And importantly, they are not photoreversible.
Why not photoreversible?
Because the amount of PFR needed to trigger the response is so minuscule, even a flash of far red light can't convert enough PFR back to PR to switch it off completely. Some seed germination examples fall here.
Okay. What about LFRs?
LFRs need more light, more like typical daylight levels. They saturate at higher light levels than VLFRs, and crucially, they are photoreversible. Red light turns them on, far red light turns them off. The lettuce seed example is a classic LFR.
Right, the red far red switch.
And for VLFRs and LFRs, generally the law of reciprocity applies. This means the response depends on the total amount of light, the fluence, not necessarily whether it was bright light for a short time or dim light for a longer time. Total dose matters.
Okay. And HIRs, high irradiance.
These are different. They need prolonged or continuous exposure to high-intensity light. The response is often proportional to the irradiance. How bright the light is, not just the total fluence. Reciprocity doesn't apply, and they aren't typically photoreversible in the classic sense. Things like making anthocyanin pigments can be HIRs.
So different light levels trigger different kinds of phytochrome responses. And you mentioned there are different types of phytochrome proteins too, Phi A, B.
Yes, in Arabidopsis, the model plant, there are 5 Phi A through Phi E. They have specialized jobs. Phi A is really important for those VLFRs and also for sensing continuous far red light, like in the shade. It's key for seedlings emerging in dim light and involved in shade avoidance. It's also light labile, meaning it gets degraded when it's in the active PFR form.
Okay, Phi A for very low light and far red. What about Phi B?
Phi B is the main player for many of those classic LFRs, the red far red reversible responses. It's critical for de-ethylation, stopping that spindly growth, greening up. It's more stable in light than Phi A. It's the key sensor for the RFR ratio in shade avoidance.
And the others, C, D, E.
They play roles too, often overlapping or fine-tuning responses, involved in things like petiole elongation and flowering time. Their specific functions often become clear when you look at plants missing multiple phytochromes. So there's a whole team, a division of labor within the phytochrome family.
Okay, so phytochrome gets activated by light, moves to the nucleus. How does it actually change gene expression? What's the mechanism?
Well, a really important mechanism involves getting rid of proteins that act as brakes on light-dependent growth. The chapter introduces a family called phytochrome interacting factors, or PIFs.
PIFs.
You can think of PIFs as dark growth promoters. In the dark, they're active, and they basically tell the seedling, keep stretching, stay pale, don't make leaves yet, find the light. They turn on genes for etiolated growth and turn off genes needed for light growth.
So they keep the plant in dark mode.
Exactly. But when red light hits phytochrome and converts it to the active PFR form,
which then goes into the nucleus.
Right. In the nucleus, PFR physically binds to these PIF proteins, and this binding targets the PIFs for destruction by the cell's protein recycling machinery, the proteasome.
Whoa. So the light signal via PFR literally gets rid of the proteins that were promoting dark growth.
Precisely. PFR triggers the rapid degradation of PIFs. By removing these repressors of light growth, the pathways for photomorphogenesis making chlorophyll, expanding leaves, are unleashed. It's a very neat way to switch developmental programs.
That's really cool. Getting rid of a repressor turns things on. Is that the only way?
It's a major way, but there's another key player involved in this protein degradation control, a complex called CoPET-EFS, and a central protein within it called CoP1.
CoP1, okay, another acronym.
CoP1 is crucial. Think of it as a master repressor of photomorphogenesis, especially in the dark.
So another break.
Sort of, yes. In darkness, CoP1 hangs out in the nucleus and it acts like a tagger. It finds proteins that promote light-dependent development. A key one is called HY5, and it tags them for destruction by the proteasome.
So in the dark, PIFs are active promoting dark growth, and CoP1 is actively destroying the factors needed for light growth.
Exactly. That's why seedlings look the way they do in the dark. Scotomorphogenesis, or dark growth, is the default program, actively maintained by getting rid of light-promoting factors.
Okay, so what happens when the lights come on? How does light affect CoP1?
Light, sensed by phytochromes, and as we'll see, cryptochromes, somehow leads to the inactivation or removal of CoP1 from the nucleus. The exact mechanism is complex, but the result is that CoP1 stops tagging HY5 and other light promoters for destruction.
Ah, so light stops CoP1 from doing its repressive job.
Right, and when HY5 isn't being constantly destroyed, it can accumulate in the nucleus. And HY5 is a transcription factor. It turns on hundreds of genes needed for photomorphogenesis, chlorophyll synthesis, leaf development, all that good stuff.
So let me get this straight. Light hits phytochrome. Active phytochrome, PFR, goes nuclear. It gets rid of PAFs, the dark promoters. And light signaling somehow inactivates or removes CoP1, the repressor of light promoters, allowing HY5, a light promoter, to build up and turn on light genes. It's like a double whammy against dark growth.
That's a great way to put it. It's a very robust switch involving removing negative regulators. This regulation of CoP1 is a central theme we'll see again.
Okay, really complex but elegant control. Right, let's switch gears now to blue light. Plants respond to blue light too, right? A whole different set of responses.
Absolutely. Blue light triggers a whole suite of things. We mentioned phototropism bending towards light, but also things like inhibiting stem elongation, promoting cotyledon expansion, anthocyanin production, chloroplast movement, stomatal opening, influencing the circadian clock. It's a very busy wavelength.
And the chapter mentioned that blue light responses sometimes have a bit of a lag time, and they can persist for a while even after the light goes off.
Yes, that's often observed. Unlike, say, the instantaneous steps of photosynthesis, these signaling responses can take a little while to get going and then coast for a bit. That persistence is likely because it takes time for the activated blue light photoreceptor to switch back off, to revert to its inactive state in the dark.
Makes sense. So who are the main players for blue light? You mentioned cryptochromes and phototropins.
That's right. Let's start with cryptochromes, or CRYs. Cryptochromes are major regulators of blue light-driven photomorphogenesis. Things like suppressing hypocotyl elongation, stopping that stem stretch, promoting cotyledon expansion, making pigments, and they're also really important for regulating the circadian clock and controlling flowering time.
How were they found?
Well, one of the first ones, CRY1, was identified because scientists found a mutant Arabidopsis plant, they called it IKY4, for long hypocotyl that grew with really long stems under blue light. It clearly wasn't responding properly. The gene responsible turned out to encode a protein similar to bacterial DNA repair enzymes called photolyases, but it lacked that repair activity.
Interesting. So they sense light using what?
They use flavins, FAD, and another molecule called a pathogen, MTHF, as their chromophores. Blue light causes a change in the redox state, the electron status, of the FAD, and that triggers a shape change in the cryptochrome protein.
Similar idea to phytochrome. Light causes a shape change. And where do they act? Nucleus again.
Primarily, yes, CRY1 and CRY2 act in the nucleus. And here's where it connects back. Their main way of signaling seems to involve interacting with that COP1-SPA1 complex again.
COP1. It's everywhere.
Okay, so what do cryptochromes do to COP1? Do they degrade it?
Not quite degrade it, according to the model presented. Instead, when blue light activates cryptochrome, the activated cryptochrome protein physically binds to the COP1-SPA1 complex within the nucleus.
Binds to it.
Yes. And this binding seems to inhibit COP1's activity. It prevents COP1 from tagging those light-promoting transcription factors like HY5 for destruction.
Ah, so phytochrome signaling gets COP1 out of the way, maybe by removal or inactivation, but cryptochrome signaling ties COP1 up by binding to it.
That appears to be the main mechanism for CRY1 and CRY2. They interfere with COP1's ability to repress photomorphogenesis. So different photoreceptors sensing different colors are converging on this central COP1 hub to release the brakes on light-induced development. It's like different managers telling the same security guard, COP1, to let someone through, HY5, but using different methods. One escorts the guard away, the other distracts him.
Huh. That's a pretty good analogy. And it's worth noting CRY2 can also directly bind to certain transcription factors to regulate specific processes like flowering time.
It's really clear these systems aren't working in isolation, though. The chapter talks about coaction.
Yes, definitely. Photoreceptors interact and cooperate. They integrate signals from red, far red, and blue light to fine-tune the plant's responses. Stem elongation is a great example. Both red light via phytochromes and blue light via cryptochromes and phototropins inhibit stem growth.
But maybe at different speeds or under different conditions?
Exactly. Experiments using different light backgrounds or looking at the timing show that blue light often causes a very rapid, almost immediate decrease in elongation rate, while a phytochrome effect might take a bit longer to kick in. Mutant analysis helps tease this apart too, showing which receptor is needed for the initial rapid phase versus the later sustained inhibition.
And things like flowering time and the circadian clock are prime examples of this cooperation, right?
Absolutely. Setting the internal clock and deciding when to flower involves complex interactions between multiple phytochromes and cryptochromes, integrating information about day length, light quality, and light intensity.
Okay, let's move to the other key blue light sensors, the phototropins, or PHOTs.
Right. These are the primary receptors for phototropism, the directional bending towards blue light.
How are they discovered? More mutants.
You guessed it. Mutants that failed to bend towards light. They were called N-phi-8 mutants for non-phototropic hypokidot. Plants typically have two main ones, PHOT1 and PHOT2, and they're located right at the cell surface, in the plasma membrane.
Not in the nucleus this time.
Correct. PHOT1 is particularly important for sensing low intensities of blue light for phototropism, while PHOT2 kicks in more strongly at higher intensities. But they both contribute to other blue light responses too, like chloroplast movements and stomatal opening.
What kind of proteins are they?
They are light-activated enzymes called kinases, specifically seronephronine kinases. They have these specialized light-sensing domains called LOV domains. LOV stands for light oxygen voltage sensing, and each LOV domain binds a flavin chromophore, FMN this time.
LOV domains. Okay, so blue light hits the LOV domain.
When blue light hits the FMN chromophore within the LOV domain, it causes a temporary covalent bond to form between the FMN and a part of the protein itself, a cysteine residue. This chemical change triggers a significant shape change in the LOV domain and the protein regions connected to it.
And that shape change activates the kinase part.
Exactly. In the dark state, the N-terminal part of the protein containing the LOV domains is thought to kind of cage or inhibit the C-terminal kinase domain. Blue light absorption by the LOV domains, especially LOV2, causes this structure to unfold or rearrange, releasing the inhibition on the kinase domain.
Like pulling a safety pin.
Pretty much. This allows the kinase to become active, and the first thing it does is phosphorylate itself autophosphorylation. It adds phosphate groups to specific serine residues on the phototropin protein. And this autophosphorylation step seems to be absolutely essential for all the downstream responses mediated by phototropins.
Okay, so light activates the phototropin kinase via this LOV domain, shape change, and autophosphorylation. What does the active kinase then do? What does it phosphorylate?
Well, for phototropism, the signal ultimately leads to changes in auxin transport across the stem, creating that differential growth that goes way back to Darwin's early experiments. But phototropins also directly control chloroplast movements within the cell.
Moving chloroplasts. Why?
To optimize photosynthesis. In low light, they move the chloroplast to the cell surface to capture as much light as possible. That's the accumulation response. But in really strong, potentially damaging light, they move them away to the sides of the cell to avoid getting fried, the avoidance response. Phototropins regulate the actin cytoskeleton that moves the chloroplasts. PHOT2 is particularly key for the avoidance response.
That makes sense.
What else?
Stomatal opening. Phototropins are the primary blue light receptors that signal guard cells to open the stomata, those little pores on the leaf surface.
Right, for CO2 uptake. How does that work?
It's a really well-understood pathway now. Blue light activation of PHOT1 and PHOT2 in the guard cells triggers a signaling cascade. This cascade ultimately leads to the activation of a proton pump, the plasma membrane H+ ATPase in the guard cell membrane.
The pump that pushes protons out.
That's the one. Pumping protons out makes the inside of the cell more negative, which drives the uptake of potassium ions, K+, through channels. Water follows the potassium by osmosis, the guard cells swell up, and the pore opens.
And phototropin activates that pump.
Yes, the signaling pathway triggered by phototropin leads to the phosphorylation of the H+ ATPase pump itself, specifically on its C-terminal end. This phosphorylation allows other proteins, called 14-3-3 proteins, to bind to the pump, and this binding locks the pump in its active proton-pumping state.
Wow, that's quite a detailed mechanism. And does this interact with other signals like drought?
It does. For example, the drought stress hormone, abscisic acid, or ABA, can inhibit this blue light pathway, partly by interfering with the dephosphorylation steps needed to activate the pump. This helps the plant keep stomata closed and conserve water during dry conditions. It shows how light signals are integrated with other environmental cues.
Okay, amazing detail there. Lastly, the chapter covers ultraviolet B radiation, UVB.
Right. UVB can be harmful, damaging DNA, but plants also use it as a specific signal to trigger protective responses and some developmental changes. like making flavonoid sunscreens or affecting leaf growth.
And it has its own photoreceptor, not phytochrome or cryptochrome.
Correct. The specific UVB photoreceptor is called UVR8, UV resistance locus 8. It's structurally very different from the others. It's a protein composed of repeating units that fold into a shape like a propeller with 7 blades. In the dark, it exists as an inactive pair, a homodimer.
Okay, a dimer like phytochrome. Does it have a chromophore?
And here's the really wild part. No, UVR8 doesn't have separate non-protein chromophore like the others.
What? How does it sense UVB then?
It uses some of its own amino acids. Specifically, tryptophan residues within the protein structure itself act as the primary UVB sensors. They absorb the UVB photons directly.
The protein itself absorbs the light. That's incredible.
It is. And this absorption by the tryptophans causes specific bonds, salt bridges, holding the two monomers together in the dimer to break. This breakage causes the dimer to dissociate, to fall apart into two active monomers.
So UVB splits the inactive dimer into active monomers. How do the monomers send the signal? Don't tell me, CoP1 again.
You guessed it. The UVR8 monomers move into the nucleus and interact with the CoQ1 SPA complex.
Okay, but wait. Phytochromes and cryptochromes signal to inhibit CoP1's repressive function in light development. What does UVR8 do with CoP1?
Here's the twist. In the UVB signaling pathway, the interaction between the UVR8 monomer and the CoP1 SPA complex seems to activate CoP1's function, or perhaps redirects it.
Activate it. CoP1, the thing that stops light growth in the dark, is now being activated as part of a light response pathway.
It's a fascinating switch in roles. In this context, the UVR8 CoP1 SPA complex acts as a positive regulator. It promotes the activity or stability of transcription factors like HY5.
HY5 again.
Which then turns on the genes needed for UVB protection, like flavonoid biosynthesis enzymes.
That is absolutely wild. The same protein complex, CoP1 SPA, acts as a brake for red-blue light-induced growth, but as an accelerator pedal for UVB protective responses, all depending on which photoreceptor upstream is talking to it.
It highlights the incredible modularity and, frankly, the complexity of these signaling networks. Proteins like CoP1 are central hubs, and their output is completely reconfigured depending on the input signal. The chapter also mentions other proteins, RUP proteins, that help put the brakes on by encouraging the UVR8 monomers to redimerize and become inactive again once the UVB signal stops.
Wow. Okay, what an absolutely amazing deep dive into how plants sense and respond to light. They really aren't just passive green things, are they? They have these incredibly intricate molecular systems, phytochromes, cryptochromes, phototropins, UVR8, each acting like specialized eyes, sensing different colors, different intensities, different directions.
And these systems regulate just about everything, from a seed deciding whether to germinate, to a seedling completely changing its shape when it hits the light, controlling which way it grows, setting its daily rhythms, making sure photosynthesis is efficient, and even protecting itself from sunburn. It's fundamental.
So what does this all mean?
I think it means that what looks like a simple process, a plant growing or flowering, is actually the result of a highly sophisticated ongoing computation of the light environment. Each photoreceptor, its specific location in the cell, the chromophore it uses, its stability, the proteins it talks to, all these properties, allow the plant to extract incredibly precise information from just one environmental factor, light. And the crosstalk between these systems, sometimes, as we saw, using the very same components like COMP1 in completely opposite ways, well, it allows for this remarkably versatile and finely tuned set of responses. It maximizes their chances of survival and success. It's like they're constantly reading this complex language written in light.
Absolutely fascinating. And I think this deep dive really has taken us through the key mechanisms, the developmental changes, the molecular players, the experiments, all laid out in that chapter, really showing how plants use light, not just as energy, but as crucial information.
A hidden world definitely revealed.
Well, thank you so much for joining us on this deep dive into how plants quite literally see the light.
My pleasure. Until next time.